Welcome to this video. In this video I'm going to explain to you how it is that the folate antagonist antibiotics actually achieve a bacteriostatic effect. And don't worry, if you don't know what bacteriostatic is, I will define that later on. Okay, so let's begin by talking about the two different classes then of folate antagonist antibiotics. So we're going to discuss both of these. So class number 1 is a large class of antibiotics known as the sulfonamide antibiotics. Okay, so the sulphonamides, also known as the sulfur drugs, and we'll discuss these ones first. Okay, so there's a large class of the sulfonamides which were all, are used or were used as antibiotic chemotherapy agents. Okay, and we'll discuss those and how they work. They are folate antagonist antibiotics. The second class of folate antagonist antibiotics only contains one drug, okay, and this is the drug trimethoprim. Now, these two different classes then, sulfonamides and trimethoprim, they both are going to take effect against bacteria by blocking the activity of folate. Sulfonamides, as we'll see, will block the synthesis of folate. Trimethoprim will block the action of folate. Okay, and by blocking the action of folate, they're going to be able to block the bacteria from synthesizing the nucleotides that they need to be able to synthesize to make DNA and RNA. And if the bacteria can't make DNA and RNA because they haven't got the correct nucleotides, then of course they can't make proteins because making RNA is a prerequisite to being able to make proteins, and they can't divide because being able to make DNA is a prerequisite to being able to divide. If you're going to divide, you're going to have to copy your entire genome. So if you can't make DNA, then you can't divide. So they're going to be able to stop cells from dividing. Also, of course, being able to make proteins is essential for being able to divide because you need to have twice as many proteins if you're going to become two cells. Okay, but we'll go over that all again later. So let's begin then with the sulfonamide antibiotics, and then we'll go on to trimethoprim. So let's start with class 1, the sulfonamides. So the starting point then to ask is, what actually is a sulfonamide? So a sulfonamide, just looking at the name, you'll be able to guess that this categorization of antibiotics is a chemical categorization. Okay, sulfonamide, we are categorizing a bunch of drugs and putting them all together because they have the same chemical structure. And that chemical structure that they've all got within them is the sulfonamide functional group. So the first thing I want to explain to you is what is the sulfonamide functional group. So we'll start off with what is sulfonic acid, and then from that I will motivate what a sulfonamide is. So let's have a look at what the sulfonic acid functional group is. So the sulfonic acid functional group is a functional group that lots of different molecules can have as part of them. And this is what it looks like. It has a sulfur atom at its centre, as you might expect, and then it's got two oxygens that are double bound to it, like so, okay, and then it will also have an alcohol group coming off it, like so, and then it's this remaining bond here, that's the free bond, that will then attach it to whatever molecule it's attached to. So I'll put the R group would be here, okay, where R can be some arbitrary other other chemical structure. So this is a functional group then. The sulfonic acid functional group is what we've got sitting in front of us. And this is of course an acid because this alcohol group here, it's capable of donating its proton away. And really you should compare this to, I'm sure, what will be a more familiar chemical structure to you, which is the carboxylic acid group. Okay, so here, this is the carboxylic acid group where we have a carbon double bound to an oxygen with an alcohol group attached to it. Okay, so for instance, this is the group that's in methanoic acid, benzoic acid, ethanoic acid, which is in vinegar. All of those much more familiar acids, they all have this functional group, this carboxylic acid functional group. But really, this sulfonic acid functional group, the reason it's an acid is very similar to the reason that this is an acid. The reason is that this alcohol group here is capable of donating a proton away. So if I just draw this bond out in a little bit more detail, in the alcohol group we have this single bond between the oxygen atom and the hydrogen atom. So the oxygen atom has put an electron into this bond, and the hydrogen atom has also put an electron into this bond. Again, they're sharing the pair of electrons in between them here in this covalent bond. Now, what of course can happen is that the rest of the hydrogen atom, and remember hydrogen is a very simple atom, it's made up of just a proton with an electron orbiting it. Okay, so once it's put its only electron into this double bond, the only portion remaining of the hydrogen atom is the proton. So the hydrogen nucleus, the proton, can decide to go off. It can cleave away from here and it can abandon its electron with the oxygen atom, and that then gives the proton away, donates the proton away into the solution. Okay, and it's that donation of protons into free solution that defines acids. So acid molecules are molecules which are capable of donating protons away into solution. So that's why the carboxylic acid functional group here is indeed an acid, because it can donate this proton away into solution and the oxygen will then be left with a negative charge. It's also why this sulfonic acid functional group is actually an acidic functional group, because it can donate its proton away and leave the electron on the oxygen atom with a negative charge. Okay, so that's the sulfonic acid functional group, which is present in many different molecules. So now let's use this to motivate the concept of a sulfonamide functional group. So now what is a sulfonamide? Well, this is analogous to the concept of an amide with a carboxylic acid functional group. So again, I want to appeal to chemical understanding, organic chemistry that you will actually know. So you're more likely to have met carboxylic acids and amides than you are to have met sulfonic acid and sulfonamides, but sulfonamide is equivalent now to the concept of an amide, just as the concept of a sulfonic acid functional group was equivalent to the concept of a carboxylic acid group. So what we're going to do is we're going to bind onto this acidic sulfonic acid functional group here an amine. Okay, through a condensation reaction we're going to take the alcohol group off, take a hydrogen off the amine group, and then bind the nitrogen to the sulfur. Okay, so going back to an amide which is the equivalent for a carboxylic acid binding to an amino group, what you would do is you take the alcohol group off the carboxylic acid group, you take a hydrogen off the amino group, and you then bind the nitrogen of the amino group to the carbon atom, and then you could have other functional groups coming off here. So you could have R2 and R3. Okay, if I now replace this one with R1, and this then is the concept of an amide link. So this link here is known as the amide link, and this entire functional group that you could have within your molecule then is known as the amide functional group. Well, a sulfonamide is the equivalent, but with the sulfonic acid functional group rather than the carboxylic acid functional group. So let's do this. Let's take the alcohol group off the sulfur atom, take a hydrogen off the nitrogen, or potentially, well, it will need to be a hydrogen. Okay, take the hydrogen off the nitrogen atom and bind this nitrogen atom to the sulfur atom, and you'll end up with a sulfonamide. So let me now draw this here. So here's the sulfur atom in the middle with two oxygen atoms still double bound to it. We'll have the first R group here, which I'll now label up R1, and then we'll have a nitrogen atom here with two other functional groups, R2 and R3, attached to it. And this functional group then, this is what is meant by a sulfonamide. Okay, so all of these sulfonamide folate antagonist antibiotics then, they are all going to contain one of these functional groups. In fact, I will now circle this round because this is the important bit. All of the rest was just motivating this structure. This is the important functional group then that all of the sulfonamide antibiotics, all of the sulfur drugs are going to contain within them. Okay, right, so now let me show you some notable examples then of sulfonamide antibiotics, just to completely get this into your head. So I'm actually going to show you the chemical structures of these sulfonamide antibiotics, and the major thing that I want to show you is that they all contain this functional group. Now, the first thing to say is that there are a huge number of sulfonamide antibiotics. Very few of them are now still used clinically, and the main problem for the use of the sulfonamide antibiotics is that there is a huge amount of antibiotic resistance out there to them now, and I will discuss the mechanism by which bacteria can acquire resistance to sulfonamide antibiotics later on. Okay, so not many of the sulfonamide antibiotics are now still used, so we're not going to actually go through that many examples. The examples that I'm going to go over are, I'm going to give you sulfanilamide, because that's was the first sulfonamide antibiotic, and then I'm also going to mention the two that are still clinically used often, which is sulfamethoxazole and also sulfadiazine. Okay, so let me now go through some examples then of sulfonamide antibiotics. So the first example that I said I was going to go through is the first example that actually came to be. So the first example of a sulfonamide antibiotic, the first one to actually be used, and this is sulfanilamide. So sulfanilamide, and what I want to do is I want to show you the structure of sulfanilamide, and really the main purpose of this is just to convince you and nail into your head that they are all going to contain the same functional group, which is the sulfonamide and sulfonamide functional group, and that's the reason that they're all called sulfonamides. Okay, so sulfanilamide, very simple structure. It is in fact going to have R2 and R3 here, these two R groups, just as hydrogens, so that makes it much more simple. And then all we're going to have that's more complicated is this R1 group here, which is going to be a benzene ring with an amino group coming off it as well. Okay, so let me now draw you out the structure of sulfonamide. So I'll start by putting in this benzene ring. So of course a benzene ring is a ring of 6 carbon atoms, where each of the carbon atoms has a bond, a bond to another, to 2 other carbon atoms, and then they have donated an electron into a delocalized ring of electrons. So I'll put the delocalized ring here, and they usually, in pure benzene, all of the carbons also have a hydrogen atom coming off them. Okay, of course, not all of them are going to actually have hydrogens coming off in our structure, because some of them will have bound instead to other things. Okay, so now let me put the sulfonamide functional group coming off here. So I'll have the sulfonamide functional group here. So here's the sulfur atom, double bound to 2 oxygens, and then we'll have our nitrogen here with just 2 hydrogens coming off there. Okay, and now the one extra thing that we're going to stick on here is an amino group, which is going to be opposite the sulfonamide group here. Okay, so we're going to have an amino group here. And this then is the structure of sulfanilamide, and as I say, this was the first sulfonamide antibiotic to actually be discovered to have antibacterial properties, and it was the first one to be used. Okay, so that's why it is a notable example to know. It's not very commonly used anymore. Okay, so now that we've had the first one, let's now do the 2 clinically important ones, because they're still reasonably commonly used. Okay, so the main example of a sulfonamide antibiotic that's still used is sulfamethoxazole. Okay, and now I'll show you the chemical structure of sulfamethoxazole. Okay, and by the way, sulfamethoxazole, it's often abbreviated down to SMX. So if you see people referring to SMX, particularly as we'll see when they're writing a combination, a drug that contains a combination of sulfamethoxazole with trimethoprim, they often write it just as SMX, like so. So sulfamethoxazole or SMX, both of those refer to the same thing. Okay, so now let's actually have a look at the structure then of sulfamethoxazole. So once again, we want to see that it's going to contain this sulfonamide functional group. Okay, and in fact, it has a very similar structure to sulfanilamide. It's really just a more complicated version of sulfanilamide. In the R1 position, it's going to have the exact same functional groups. I'll start off just by replicating all of what we've got for sulfanilamide here. So here is the benzene ring, like so, with its delocalized ring of electrons. And then we're going to have the sulfonamide functional group here, the sulfur double bound to 2 oxygens, and then the amino group coming off here. And now we're going to make it more complicated because of in one of these R positions here, it doesn't matter whether you want to see it as R2 or R3, we're going to now have a more complicated group coming off. So we've got one hydrogen, but now we're going to have a more complicated functional group coming off here, and this is the methoxazole portion. Okay, and it's actually not going to contain an oxazole ring, it's actually going to contain an isoxazole ring. So let me now draw out the ring that we're going to have here. So we're going to have what is known as an isoxazole ring, and I'll explain what the difference between an isoxazole ring and an oxazole ring is in just a moment. So firstly, let me just draw out the structure of an isoxazole ring. So an isoxazole ring is a 5-membered ring where 3 of the members are going to be carbon, 1 of the members is going to be oxygen, and the other is going to be nitrogen. Okay, so let me draw this out. So here is a carbon, here's a carbon, here's a carbon, here's a nitrogen, and up at the top then we'll have an oxygen like so. And we also need to put some double bonds on here, so we're going to have a double bond there and a double bond there. Okay, and then if you were completing this as a pure isoxazole ring, you'd have a hydrogen coming off this carbon, a hydrogen coming off this carbon, and a hydrogen coming off this carbon, and that would then be called an isoxazole ring. Now what is the difference then between that and an oxazole ring? Well, an oxazole ring would be this structure here. You'd have an oxygen at the top, like so, and then you'd have a carbon on either side of the oxygen, and then you'd have the nitrogen down at the base rather than next to the oxygen as it is up here. But again, you'd have double bonds in these 2 positions like so, and you'd have hydrogens coming off all of the 3 carbons to take their bonds up to 4 each. Okay, so this structure would then be the oxazole ring. Okay, now you might be wondering about its name, oxazole. Ox is clearly for oxygen here. What does azole mean? Well, an azole ring in chemistry, just to give you a bit of background into the organic chemistry, an azole ring is a 5-membered ring where 3 of the members are carbons. Okay, so in any azole ring you'll always have 3 carbons here. Okay, 1 of the members is a nitrogen. You're going to have 2 double bonds in the ring, so I'll colour those in as well. And then the final member of the ring can either be a nitrogen, or it can be an oxygen, or it can be a sulfur. So in the case of an oxazole ring, we are talking about an azole ring where the final member is an oxygen. If we're talking about a thiazole ring, so if I put phi in front of there, thiazole ring, that would be a sulfur, so you'd replace the oxygen with sulfur, so phi generally denotes things to do with sulfur. If we had imide in front of there, to make an imidazole ring, we'd have a nitrogen atom there. Okay, imide generally implies something to do with nitrogen. Of course, if we had nitrogen there, nitrogen likes to have 3 bonds, so you'd also have to have a hydrogen coming off the nitrogen. Okay, so those are the 3 different types then of azole ring. And around this thing here, this atom that is, if you like, the top of the pentagon, the atom that's the one that can change, either the oxygen, the sulfur, or the nitrogen, you need to have 2 carbons linked to that, and the other nitrogen that's always there needs to be at the bottom. An isoxazole ring is an oxazole ring where we have moved the nitrogen up so that it's actually next to the oxygen. Okay, and ice, of course, means that it's going to be a structural isomer of the oxazole ring. So, strictly speaking, in sulfamethoxazole you do not have an oxazole ring. It should be sulfamethisoxazole. However, someone who came up with the name of this was being kind, so they got rid of the isoxazole and just had oxazole. Okay, now where does the meth bit come from? Well, this structure isn't complete. You're going to have a methyl group coming off here, which is then going to make this methoxazole, or methisoxazole if we're being strict. Then, just to finish this, we'll have a hydrogen coming off that carbon to take up its bond number to 4. So this now is the structure of sulfamethoxazole, which is the main clinically used sulfonamide antibiotic. Okay, right, there's one more that I want to show you, another one that is still clinically used. Not as important an example as sulfamethoxazole, but still an important one to know, which is sulfadiazine. Okay, and again, what we're going to see is that, again, it's going to be based on sulfonilamide. It needs to have this benzene ring in it with the amino group, and we will see that having that benzene ring with the amino group coming off the sulfur atom here is extremely important for the ability of these drugs to actually have an antibiotic effect, to actually have some sort of effect on the folate pathways within bacterial cells, and we'll understand exactly why that is very shortly. Okay, so for now, let's just go through another example of, and this is the final example I'm going to do, of a sulfonamide antibiotic. Okay, so firstly, let's just start by copying out the core structure, the structure from sulfonilamide here. So we've got our benzene ring here, like so, with its delocalized ring there. Then what we'll have is the sulfonamide functional group here, so the sulfur double bound to 2 oxygens. Then we'll have the nitrogen with its hydrogen coming off it. And now, of course, just like we did with sulfamethoxazole, we're going to make things more complicated. We're going to put something more interesting coming off this nitrogen here. And what we're going to actually put on is something known as a pyrimidine ring. Now, a pyrimidine ring is another ring structure that you should be aware of from organic chemistry. And the reason you should be aware of it is because it is the structure on which the two nucleic acid organic bases of DNA, thymine and cytosine, are based on. So the reason thymine and cytosine are referred to as the pyrimidines, you might have heard people referring to the pyrimidine organic bases, which are cytosine and thymine. The reason those are called the pyrimidine organic bases is that their structure is based on the structure of the pyrimidine ring. Okay, so this is a structure from organic chemistry that biologists really should be aware of. Now, what is then the structure of the pyrimidine ring? Well, it's really a modification of the benzene ring. In the benzene ring, you have a delocalized ring of electrons where every single one of the atoms in this 6-membered ring has put in an electron into that delocalized ring. Okay, and then all of the carbons, as well as having 2 bonds to other carbon atoms in that 6-membered ring, they also have a bond to a hydrogen atom. In a pyrimidine ring, you're going to have a 6-membered ring, but not all of the atoms are going to be carbon in this case. Okay, 2 of them are going to be nitrogens. So you're going to have 2 nitrogens, and they're going to be separated by a single carbon, like so. So this is how we would draw the pyrimidine ring. So we've firstly got our ring of 6 atoms, where each one of the atoms in the 6-membered ring is bound to 2 other atoms. Okay, but this time we have replaced 2 of the carbons that we would have had in the benzene ring with nitrogen atoms here. Now, in reality, what's going to happen is each one of these is going to put an electron into a delocalized ring. However, with regards to pyrimidine rings, we don't draw it in the way we draw benzene with the actual ring like this. Instead, what we do is we denote it with alternating double and single bonds, which I'm sure you will have seen in pictures for benzene. Sometimes people, instead of putting the ring, they put alternating double and single bonds. That was an older model for the structure of benzene before we really understood that actually the electrons are just in this delocalized ring in the middle of the ring of atoms. Okay, it's the same for the pyrimidine ring. The electrons are not really in double and single bonds that alternate. Instead, they are in a delocalized ring, but we still show it as alternating double and single bonds, like so. Okay, and then of course you'd have hydrogen atoms coming off each of the carbons, but you wouldn't need a hydrogen coming off the nitrogens now because they only need 3 bonds. Okay, right. Then what we're going to do is we're going to attach this pyrimidine ring. So this, by the way, this is the structure of the pyrimidine ring. This is the pyrimidine ring. Okay, this is the structure on which adenine and thymine, those 2 important organic bases in the structure of DNA, are based. We're now going to put this onto our nitrogen here. Specifically, we're going to attach this carbon here to the nitrogen. Okay, and I'll do it in skeletal. Well, actually, I'll just be small. And I was going to do it in skeletal structure to make it fit in, but I'll just do it small, like so. So here is our 2 nitrogens. Here are more carbon atoms, like so. And then we'll put the alternating double and single bonds in. Okay, and then we'll attach some hydrogens on here just to take everything up to the right number of bonds. Okay, and there now, I hope you can see it, is the structure of sulfadiazine. Okay, so we've now seen these 3 examples then of sulfonamide antibiotics: sulfanilamide, sulfamethoxazole, and sulfadiazine. Okay, right. What I now want to turn my attention onto is how do they actually work. We've seen examples of what they actually look like. We've seen that they all have this sulfonamide functional group in them, and that's the reason they're all referred to as the sulfonamide antibiotics. But we want to know, how do they actually stop bacteria dividing? And by the way, that is what is meant by a bacteriostatic antibiotic. So we can classify antibiotics as either being bacteriostatic or bactericidal. Okay, these are adjectives that we can apply to every antibiotic. We can either call an antibiotic bacteriostatic or we can call it bactericidal. Okay, now bacteriostatic means that the effect that it has on the bacteria is to stop them dividing. Okay, so they stop the bacterial cells from dividing. Bactericidal means that they actually kill, and this pen is really awful, I'll go to pink instead, means that they actually kill the bacterial cells. So bacteriostatic and bactericidal, so these ones just stop cell division. Okay, now you might wonder, well, what's the point of that? We want to actually kill the bacterial cells, don't we? We want antibiotics to be bactericidal. Okay, but if you stop cell division, then you stop the population of bacterial cells getting any bigger, and the idea then is your immune system has the chance, it has the time that it needs to actually kill the bacterial cells. So using a bacteriostatic antibiotic absolutely relies on the person's immune system being up to the job of actually clearing the cells now that it's got them under control. So the antibiotic will get the population number under control, it will stop the population number from getting any bigger, and then you rely on the immune system clearing the ones that are already there. Okay, so it is a good strategy. I mean, stopping the population getting any bigger is a good thing to do. Bactericidal antibiotics, these are slightly more powerful. They are actually going to kill the cells that are already there. Not only will they stop them dividing, they'll actually kill the ones that are already there. So if you've got someone whose immune system is very, very weak and isn't going to be capable of actually killing the bacterial cells that are already there, you would want to give them a bactericidal antibiotic rather than a bacteriostatic antibiotic. All of these sulfonamide antibiotics, and indeed trimethoprim as well, in fact all of the folate antagonist antibiotics that are the topic of this video, are just going to be bacteriostatic. They're going to stop cell division, but they're not actually going to be able to kill the bacterial cells that are already there. They just stop the bacterial cell population getting any bigger. Now, how do they actually do this? Well, they are going to inhibit an enzyme within bacterial cells. So they're going to go inside the bacterial cell. So let's just draw our little bacterial cell here. So let's say this blob is going to represent our bacterial cells. So our sulfonamide antibiotic, or indeed our trimethoprim, but at the moment we'll just stick to the sulfonamides, they are going to go inside the cell, okay? And inside the cell they are going to inhibit an enzyme which is known as dihydrotauroate synthetase. So here is this little enzyme drawn inside our bacterial cell's cytoplasm, and I'll have it in blue here. And for short, it is abbreviated down to DHPS. Okay? And I will write out its full name now. So DHPS stands for dihydrotauroate synthetase. So the DH is for dihydro, the P is then for tauroate, and then the S is for synthetase, so dihydrotauroate synthetase. Now, what does dihydrotauroate synthetase actually do, and why does inhibiting it lead to the cell no longer being able to divide? Well, dihydrotauroate synthetase is involved in a biochemical pathway that overall synthesizes a molecule known as folic acid, which is, I'm sure, a molecule that you will have heard of before. Maybe because you know that pregnant women need to take folic acid when they're pregnant, or should take folic acid when they're pregnant. Okay, you might also know that it's one of the B vitamins, specifically it's called vitamin B9. Now, not only is it a very important molecule in human cells, but it's also a very important molecule in bacterial cells. So bacterial cells value this stuff a lot as well. Okay, and they have synthetic pathways for making folic acid. Okay, and this synthetic pathway involves this enzyme dihydrotoroate synthetase. So dihydrotoroate synthetase is an enzyme involved in the synthetic pathway for making folic acid. What we are going to do if we inhibit dihydrotoroate synthetase is we're going to stop the bacterial cell being able to make folic acid. And if it can't make folic acid, what then happens? Well, we need to know, what is folic acid so useful for? Why do cells require it so much? Well, this actually goes for both bacterial cells and human cells. Folic acid is an important molecule needed for the function of the enzymes which make nucleotides. So DNA and RNA, you will be aware that those are made up of nucleotides. Remember, a nucleotide is an organic base with a ribose or deoxyribose sugar with a phosphate group. So let me just draw a little picture of a nucleotide here. So here is the sugar, which will be ribose in the case of RNA or deoxyribose in the case of DNA. Okay, then we'll have the phosphate group coming off the fifth carbon of the ribose or deoxyribose sugar, and then here we'll have the organic base. Okay, so this is the structure of a nucleotide here. So organic base, and this is the phosphate group. Okay, so in order to make DNA and RNA then, you need to have lots of these nucleotides for each of the 4 different organic bases. Now, for some of them, folic acid is absolutely essential for their production. Okay, so to produce these nucleotides that you need to make DNA and RNA, you need to have the folic acid. Now, folic acid is not actually a molecule that's going to end up as part of the nucleotide. It's not a building block of a nucleotide, but it's something that the enzymes which build these nucleotides must have in order to function. Okay, so the enzymes which are going to build, and this pen really is awful, I need to put that aside so that it doesn't come back. Okay, so the enzymes which build these nucleotides, they use the folic acid when they're actually building the nucleotides, but they don't use it as a building block. Okay, they just require it in order to build the nucleotide. So folic acid is absolutely essential for the function of the enzymes which produce nucleotides. So if you run out of folic acid inside your bacterial cell, or indeed inside your human cell, but we're talking about bacterial cells here, if we block the dihydrotoroate synthetase inside the bacterial cells and their folic acid levels therefore go down, then these enzymes which synthesize the nucleotides are no longer going to be functional, and therefore their nucleotide levels are going to go down. So are these bacterial cells going to be able to now create DNA or create RNA? The answer is no. And creating DNA and RNA are absolutely essential for cell division. Okay, creating RNA is essential for protein synthesis. Creating DNA is obviously essential for genome replication. When a cell divides into 2, it needs to copy all its proteins and it needs to copy its genome. If it can't synthesize proteins and it can't synthesize DNA, it's not going to be able to do those. So that is why blocking dihydrotoroate synthetase, which is exactly what the sulfonamide antibiotics do. So I've just put, this is what my sulfonamide antibiotic is going to do, is going to actually result in the bacterial cell no longer being able to divide. Now, what I'd like to do to add a little bit of extra information onto this pathway is actually show you why it is that all of these sulfonamide antibiotics needed to have this benzene with this amino group in it in order to be able to block the dihydrotoroate synthetase. And in order for you to understand that, we're going to have a look at the structure of the folic acid molecule. If you look at the structure of the folic acid molecule, we can get an interesting insight as to why it is that all of the sulfonamide antibiotics have this structure, this benzene ring with this amino group attached onto it. Okay, right, so let me now show you the structure of folic acid then, and I think I'll go over onto the other side in order to do this. So the structure of folic acid, it's a rather large molecule to remember the structure of. So in order to understand it, what we do is we break the molecule up into 3 separate components. Okay, so there is the pteridine ring, there is the para-aminobenzoic acid, and there is the glutamic acid. And I will start by putting the one that's going to be in the middle, which is the para-aminobenzoic acid. So right in the middle of the structure of folic acid, we're going to have what's called para-aminobenzoic acid, and this is actually the portion that is going to be incredibly necessary for us to understand in order to understand why we need to have that benzene ring with the amino group coming off it in the structure of our sulfonamide antibiotic for our sulfonamide antibiotic to actually work and inhibit dihydroteroate synthetase. Okay, so we're going to have para-aminobenzoic acid, which is often just abbreviated down to PABA for short. P for para, A for amino, B for benzoic, and then A for acid. Okay, right. So para-aminobenzoic acid, this consists of a benzoic acid molecule with an amino group opposite to the carboxylic acid group coming off the benzene ring. So let me draw this out for you. Okay, so we'll have the benzene ring in the middle, like so. And then what we're going to need is the carboxylic acid group coming off one of the carbons here. Now, because I don't just want to draw the pure para-aminobenzoic acid, I want to draw it as part of folic acid. The carboxylic acid of para-aminobenzoic acid is going to be linked to an amino group of a glutamic acid molecule by an amide link. So I'm not actually going to draw the carboxylic acid group in its pure form, but you can imagine putting the alcohol group off here and then you'd actually have the carboxylic acid group. So I'm being fortuitous, I'm not drawing the full carboxylic acid group here. Neither am I going to draw the full amino group over here. Instead I'm just going to put the nitrogen with a hydrogen like that, because again it's going to be bound to something else. Okay, but in pure para-aminobenzoic acid, what you'd have is the carboxylic acid group here, and that will create your benzoic acid. When you've got a benzene ring with a carboxylic acid group off it, that's what's known as benzoic acid. Para-amino, para means opposite, okay, so this is saying that the amino group is opposite, and you can see it's exactly opposite to the carboxylic acid group. So because it's coming off the carbon that's directly opposite the carbon that the carboxylic acid comes off, we call it para-aminobenzoic acid. And again, I haven't drawn the amino group in its fullness because it's going to be bound to something else. So if we were just drawing para-aminobenzoic acid, the pure molecule, I'd put a hydrogen off here and an alcohol group off here, but we're not, we're drawing folic acid. So this is the para-aminobenzoic acid portion of folic acid. Now let me put on the glutamic acid portion. So we're going to have the amino group of the glutamic acid residue bound by amide link to the carboxylic acid group of the para-aminobenzoic acid molecule. Okay, and glutamic acid is of course an amino acid, so it will have the classic amino acid structure. Here's the amino group, the alpha carbon in the middle, and here is the carboxylic acid group, and this carboxylic acid group will actually be in its pure form, and it's this carboxylic acid group that actually makes folic acid an acid. And then off the alpha carbon we will of course have a hydrogen, and we'll also have the R group of the glutamic acid. And the R group of the glutamic acid consists of 2 methylene groups, or we could call them an ethylene group, and then another carboxylic acid group, which will also contribute to the folic acid being an acid group. So these are the 2 acid groups that we're going to then have in our folic acid molecule. So this portion of the folic acid molecule here, hopefully you recognize this as the important protenergic amino acid, which is glutamic acid. Okay, right, so far we've seen that to make a folic acid molecule you bind a glutamic acid molecule by amide link onto the carboxylic acid group of a para-aminobenzoic acid molecule. However, we're now going to add a far more complicated chemical structure on the other side, which is known as the pteridine ring. So we're going to add on the pteridine ring now, and this is actually going to consist of 2 6-membered rings that are fused together on one side. Okay, so we're going to see our friend that we saw earlier, which is the pyrimidine ring, is one of them, but now we've got another ring, another new ring to introduce, which will be the other, and this is going to be the concept of a pyrazine ring. That's a very similar concept to the concept of a pyrimidine ring. It's just that now the nitrogen atoms are in different positions. In fact, it's pretty much the exact same concept. So again, when you remember when I was discussing the pyrimidine ring, I told you that effectively what we're going to do is we're going to do the same thing as we do in benzene, but instead of having 6 carbons, we're going to have 2 nitrogens and 4 carbons. In a pyrazine ring, we're going to have the exact same thing. We're going to do the same thing as benzene. We're going to have 2 nitrogens, 4 carbons. We're just going to have the nitrogens slightly more spaced out. Okay, so we're going to have them opposite one another, like so. And I'll draw its skeletal structure here, so I won't show the carbons. So these are obviously the carbons here. And again, we don't usually actually draw a delocalized ring, although that's really what you'd have. We put alternating double and single bonds like so. So here, these are the 2 nitrogens opposite each other. Remember, in the pyrimidine ring, the nitrogens were only separated by a single carbon. We have the 4 carbons here. Each of those would have a hydrogen atom off it. Remember, really what's happening is each one of the members of the ring is donating an electron into a delocalized ring in the center, but we don't draw that in any case except for benzene. Instead, we put alternating double and single bonds, but it's the same idea. Okay, so we're going to have one of these pyrazine rings then coming off this nitrogen here after you have a methylene group. Okay, so you're firstly going to have a methylene group here, and then you're going to have the pyrazine ring coming off here. And I will draw the pyrazine ring in skeletal form rather than in molecular form. Okay, it just makes it easier on the eye. Okay, like so, because then we don't have to bother putting all the hydrogens off the carbons. So I have alternating, and I prefer really to have the double bond there, but it doesn't actually matter. So we can have alternating double and single bonds like so. Okay. And now what it will make the diagram slightly bad. Yes, allow me just to change this. Let me again remember what I say, that the alternating double and single bonds, that's just a way of drawing it. Really what's happening is the delocalized ring, but to draw this better, let's have the alternating double and single bonds in that position. The reason is that we're going to have another one here, and it will be better to have them both sharing that double bond there, okay, because each of these carbons is only going to put 1 electron into the delocalized ring, and we're going to have delocalized rings for both of these rings, and if you don't put the double bond there, it makes it look awkward because you only want to show these putting in 1 electron. So if you don't put it there, it makes the diagram look awkward. I'll show you why in a moment. Just go with it being like this for the while being, okay? Okay, the reason, as I say, is that we're going to add another ring on here. You will see the reason in a moment. Okay, right. So now another ring then. So we're going to now have a pyrimidine ring attached onto this. So we're going to have 2 nitrogens again, but now separated just by 1 carbon. So here comes our pyrimidine ring, and you're going to have 1 nitrogen down here, 1 nitrogen up here, like so. And this is the reason I wanted that double bond there, so that that double bond is now a double bond in both of these rings. It just makes the picture look nicer. If I hadn't done it like that, then I'd have had a double bond here, a double bond here, a double bond here. Where would I have now put the double bonds here? Well, I'd have had to put them in the same positions as here, and that wouldn't have really mattered, but it just looks better to have it like this, because then look, we've got alternating double and single bonds in both of them, whereas if you had it like that, then you wouldn't have had a double bond here, and it wouldn't have made any difference, because the meaning of this, of course, is really that each one of the 10 atoms in this fused double ring system here has put an electron into some sort of delocalized structure that is spanning the entire double ringed thing here. But just because we show it with these alternating double and single bonds, that's the nicer way of drawing it in this case. Okay, so I've put the double bonds in these orange positions rather than in the crossed out positions. Okay, right, so we have these 2 fused rings then, which are aromatic with these delocalized rings. So we've got the pyrazine ring and then a pyrimidine ring here. Now, to finally make this into a pteridine ring, then, what we need to do is put an alcohol group coming off this carbon up here, and then we'll have an amino group coming off this carbon down here. Okay, and that entire structure then now, starting from the methylene group there, and then covering everything all the way up to this amino group right out here, that's then called the pteridine ring structure of the folic acid molecule. Okay, so that then is the complete folic acid molecule. So break it down into these 3 manageable chunks and you will be able to remember the structure of folic acid. Pteridine ring, para-aminobenzoic acid and glutamic acid. Now you might think, well, why on earth has he bothered to show me this? Well, the reason that I wanted to show you this is to show you specifically that it contains the para-aminobenzoic acid structure here. And in fact, one of the molecules that you use in the synthesis of this molecule, so in the synthesis of folic acid, one of the molecules that you actually use is para-aminobenzoic acid. Okay, so the pure version of this, so imagine chopping the pteridine ring off, putting a hydrogen here, chop the glutamic acid off, put an alcohol group here, that's para-aminobenzoic acid. You use that in the synthesis of folic acid. And indeed, the dihydroteroate synthetase enzyme inside our bacterial cell, this guy here, okay, I shouldn't say he, it is going to require the para-aminobenzoic acid to come in and bind to it, and then it will be able to do its part in the synthesis pathway for folic acid. These sulfonamide antibiotics, they will have a very similar moiety to the para-aminobenzoic acid. We have the benzene ring with an amino group, and then instead of obviously having a carboxylic acid group here, we have the sulfonamide group in all of them. Okay, that is capable of binding to the same portion of the enzyme, the dihydroteroate synthetase enzyme, as the para-aminobenzoic acid would, and it acts as a competitive inhibitor. It binds in that site, and now para-aminobenzoic acid cannot bind in that site. Okay, and it overwhelms the para-aminobenzoic acid. It binds more strongly than the para-aminobenzoic acid, and therefore you inhibit the enzyme. So you therefore stop the synthesis of folic acid. The cell runs out of folic acid, and therefore the enzymes which make the nucleotides for DNA and RNA, those stop functioning, and therefore you can't produce DNA and RNA. Protein synthesis and the possibility of genetic replication are completely halted, and therefore the cell cannot divide. Okay, so that's the mechanism by which the sulfonamide antibiotics actually produce a bacteriostatic effect. What I will talk about in the next video is firstly the resistance to sulfonamides, which is a big problem, and then I'll talk about the mechanism of trimethoprim.